Isabel Wheeler. It is January 21st of 2020, and this is week 2 lab exercise of the Aseptic technique and culturing microbes. I'm going to sterilize the E. coli lid real quick. Okay, now we'll take our pipette into the broth and get 25 milliliters of broth and put it in here. So here we have the E. coli with the nutrient broth. And now we're going to put the lid back on the culture. Wrong lid. And shake vigorously until the tablet dissolves. I have just finished shaking this container with the E. coli and the nutrient broth. Now I'm going to use my pipette to put the cultures back into here. Okay, now that I have done that, I'm going to take this and I'm going to use this to disinfect it again. Okay, and I've done that, and now I'm going to place the lid back onto here. Now we are on to the second one. We are using the epidermitis. I'm gonna take my pipette, make sure that it is clean from all the alcohol. I'm gonna do that twice. Shake and squeeze it out. Okay, so there's my pipette. We're gonna take off the lid of the nutrient broth. And we are going to sterilize this with the candle. Okay, and once we have done that, we're going to take the epidermitis, and we are going to do the same thing. Okay, we've done that. Now we are going to take our pipette. We're going to get some nutrient broth. And then we're going to shake the S. epidermitis until it has dissolved. I've shooken the S. epidermitis, and now I'm going to take the nutrient broth, set the candle aside, and we're going to squeeze this back into the broth. We are on our last test tube. Here's our nutrient broth. Here we have my yeast, candle to sterilize this test tube, and we're going to do that. Okay, so we've done that. Now I have my sterile pipette. Make sure everything's out of it. And now I'm going to grab some yeast and we're going to put it in the nutrient broth. Now that we have put 0.25 milliliters of yeast in here, okay, now we have our solution. with the yeast in it. And we're gonna take the candle again and we're gonna sterilize the lid, the top, before putting the lid on. Okay, so that is sterilized. Now we put the lid on. Okay, so I have prepared I poured my agar plates. I let them sit for about over an hour. I have all of my broths that I let sit for about 48 hours, and all of them have a culture growth in them. So first I'm gonna be starting out with the E. coli. I'm gonna grab one of my loops from this alcohol solution. I'm gonna take my first broth, E. coli. And sterilize the top of it. Now we're gonna be careful not to touch. And we're gonna take it to our plate and do zigzag motions. Okay, now we're gonna put the lid back on. And put my loop back in the alcohol. And take that back out. And now we're going to spread that to the neck section. Okay, and put the lid back on. And then spread that to the neck section. In zigzag motions. And put the lid back on again. And our last zigzag motion. Okay, so we're done with our first one, which is E. coli. So we're gonna put this one to the side.